Good day. This is the Professor, CBMD PhD. And today's Monday, November 7th, 2022. It's 2:29 and 28 seconds. AM Eastern Standard Time. And today's research, Anticancer Therapies. So let's hop to it here. Vitamin C may hold the key to improve efficacy of dendritic cell-derived anticancer cell therapies. Date November 2nd, 2022. Source: Joseph Carreras, Leukemia Research Institute. Researchers have recently shown that vitamin C improves the immunogenic properties of dendritic cells in vitro. Results recently made public show that treating the cells with vitamin C leads to a more consistent activation of genes involved in the immune response, mainly through DNA demethylation, a kind of epigenetic reprogramming. And this discovery may be useful to generate more potent dendritic cell-based therapies in the future. And since the onset of anti-cancer cell therapies, those that use living cells to find and eliminate tumors, many types of immune cells have been used. The best known cell therapies use lymphocytes, as in the highly successful CAR-T therapies. Recently, dendritic cells have attracted the scientists' attention, thanks to its ability to uptake and present antigens, small parts of a pathogen or a cancer cell, to the T lymphocytes and induce an antigen-specific potent immune activation. On this regard, loading dendritic cells with specific antigens to create immune memory constitute the so-called dendritic cell vaccines. And to study dendritic cells in the laboratory, researchers differentiate them from monocytes, also an immune cell, using a particular set of molecular signaling. And this differentiation is accomplished through a complex set of gene activation processes in the nucleus, mostly thanks to the activity of the chromatin remodeling machinery, spearheaded by the 1011 translocation family of demethylases, proteins that act upon the DNA epigenetic markers. Vitamin C was known to interact with several 1011 translocation proteins to enhance its activity, but the specific mechanism was still poorly understood in human cells. Researchers hypothesized that treating monocytes in vitro while differentiating into dendritic cells would help the resulting cells be more mature and active. Researchers showed that vitamin C treatment triggers an extensive demethylation at NF-κB P65 binding sites, compared with non-treated cells, promoting the activity of genes involved in antigen presentation and immune response activation. Also, vitamin C increases the communication of the resulting dendritic cells with other components of the immune system and stimulates the proliferation of antigen-specific T cells. Actually, the researchers proved that vitamin C stimulated dendritic cells loaded with antigens specific for the SARS-CoV-2 virus were able to activate T cells in vitro more efficiently than non-treated cells, showing the superiority of dendritic cell vaccines treated with vitamin C. Overall, these new findings support the hypothesis that treating monocyte-derived dendritic cells with vitamin C may help generate dendritic cell vaccines with higher performance. After consolidating these results in preclinical models and, hopefully in clinical trials, a new generation of cell therapies based on dendritic cells may be used in the clinic to fight cancer more efficiently. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.